nucleus, and that left only the mitochondria in the cytoplasm, and we swapped that nucleus with another nucleus. So now we have one nuclear genome paired with different mitochondrial genomes, and we use a two-letter code for these. The first letter is for the nuclear genome, the second letter is for the mitochondrial genome. C57 is C, C3H is H, FBB is F, and valve C is B. So the first set of mixed mice that we made have the FBB nuclear genome. That's the same one as in the polyomal little t. We crossed the mixed mice with the polyomal little t mouse. So now it's we have only the mitochondrial genome as the variable, and our data superimposed with those from the Hunter experiment. We only changed the mitochondria, kept the nuclear genome the same. We replicated the experiment with another oncogene, the HER2 oncogene, and Amanda Brinker showed similar types of effects. Carol and Vivian showed that the natural autochthonous just aging of these mice changed their tumor formation and metastasis formation as well. I'll just show you this data. It's brand new. It's newly published. Bladder cancer. It's one of the top 10 most common cancers. And I just will point out two things that we found very interesting. The C3H nuclear genome makes, when we treat with carcinogen, more bladder cancers. Bladder cancer is much more prevalent in males than females, but the C3H mitochondrial genome eliminates the protection of the female genome, whatever that is. We're still trying to find out. But so these complex things take place. Just to show you, because of time, these mixed mice show differences in effect on stroke, Parkinson's disease, hypertension, a number of other diseases, and along with tumor genicity and metastasis, but things still don't seem to add up. Everyone knows that in order to metastasize, you have to have different adhesion, different ability to invade, ability to escape the immune system, and so on. Well, the 13 proteins encoded by mitochondria cannot explain all of that because there are no adhesion genes, no invasion genes, no immune genes. So our first hypothesis was that somehow the mitochondrial genome, the polymorphisms that are in it, affect the nuclear transcriptome via changes in the DNA methylation or histone marks. This is just a summary of those experiments. This was done by multiple people. I will acknowledge them a little bit later. The key point is that less than 250 loci in the nuclear genome are changed for methylation or histone marks in their effect. So it's very selective changes. Well, in addition to that, one morning I'm waking up while taking a shower. I get my best ideas taking a shower. I hope some of you can relate to that. And it occurred to me the obvious. It's not only the cancer cells that inherit the mitochondrial genome, it's all of the stromal cells. So Amanda Brinker tackled this project. We took our mixed mice and we took tumor cell lines that have the nuclear background that matches some of the mixed mice that we have, and these cell lines have differential abilities to metastasize. And we injected those cells into the wild type or a mixed mouse. And here is just one of many experiments, but the bottom line, just to show you we have all the data, whenever we have C57 LAC6 mitochondria, metastasis is inhibited by 90%. Whenever we have C3H mitochondria, metastasis is 3 to 5-fold higher in 2 melanomas and 2 mammary carcinomas. We haven't changed the tumor cells at all. We've changed only the environment that they exist in, both at the primary site or the metastases. We haven't defined which one at this point. So at this point, a quick summary of what I've shown you, and I hope you're convinced. The polymorphisms in mitochondria contribute to metastasis efficiency by both intrinsic, by changing the methylome, histone marks and so on, as well as extrinsic ability. They're changing somehow the stroma, and they selectively modify the nuclear DNA methylation and histone marks. So how are they doing this? Well, this led us to embark, I think one of our earlier speakers talked about multi-omics. I call it OMG-omics because of the cost and the amount of data. We have over 15 terabytes of data analyzing 117 plausible hypotheses. That's right, 117. To grant reviewers, that means lacking focus and diffuse non-thinking. I'm looking at Vivek as he reviews grants. Hopefully you weren't the person who said behind us lacking focus. Anyway, we looked at a number of these things. I'm just going to show you some of the data. Obviously, I don't have time for all of it. But when we look